Yeah, I'll go back to this slide again. That's right now on being shown here. Pathobiology of Alzheimer's disease. It's Alzheimer's disease is also called dementia. Uh, go next slide, please. Okay. Yeah. The mechanism of Alzheimer's disease, as we know so far, it's a progressive disease. It starts probably early 40s, maybe in the age, but you don't really get memory loss until 60 plus. The problem is lots of unknowns from the age of 40 to 60 on. And those unknowns we still don't know because there are no symptoms. Without symptoms, there are no biomarkers before the onset of the actual disease. So it's very complicated progression, but we do know that as it progresses, we have different mechanisms that have been identified. Uh, Mitochondrial damage, oxidative stress, defective calcium homeostasis, gene mutations like ABP gene mutations, all these things do cause amyloid plaques in the brain, which causes synaptic dysfunction in the neurons, which actually precipitates as Alzheimer's disease. It's a very progressive and the degeneration mechanisms at molecular level, people are still identifying more and more. But this is the phenotypically, that's what we can see, and the disease progression. Next one, please. Next slide. So it's a very common, in fact, it's about 5.5 crores people in world, all over the world are having this disease, and more than 1 crore just in India. And that's common for ages 60 and older. Interestingly, it's more common in females. Family history may increase likelihood, but don't have much data on it. Interestingly, the incidence is prevalent in cerebral cortex and hippocampus, parts of the brain with loss of cholinergic neurons. That is especially the acetylcholine secreting neurons. Okay, so the phenotype is extensive neuronal atrophy with memory loss, personality changes, and interferes with daily life. Next one, please. Next slide. Okay. So pharmaceutical industry has been very active in producing drugs. There are about 28 drugs. To tell you the truth, none of them really enhance the memory. Yes, they are approved because they're safe to use, but do they really work for reversing the memory loss or preventing the progression of the disease? There's nothing, especially Aduhelm, which was approved last year by FDA. It's a Biogen drug, but it's a controversial therapy because a lot of the clinical pharmacology advisory board members at FDA resigned after FDA approved this drug, which means that the clinical outcomes are really statistically, there was not much of improvement in memory loss. The other drugs that are being used routinely are Aricept, Razodine, Exelon, Memantine, and they're also combined with each other some to improve the memory loss. But again, there's no perfect drug that helps in improving or preventing memory loss in Alzheimer's disease. Next one, please. So pathobiology of Parkinson's disease. This is also neurodegenerative disease. It is also mostly mitochondrial damage and neuronal damage. There's a model compound called MPTP that is typically is used to mimic the Parkinson disease and to understand the mechanisms and identifying the biological progression of the disease. Again, but the Parkinson's disease is different from Alzheimer's disease. Next one, please. So it is characterized with decreased dopamine with increased cholinergic activity. And this is unlike Alzheimer's disease, this is very prevalent in midbrain, which is basal ganglia, and specifically in substantia nigra. And at least 1 crore people in world are suffering from Parkinson's disease. Levodopa, which has actually mimics a dopamine, is somewhat effective in treating, and the other one, amantadine, which increases actually the dopamine levels in the brain, are used as standard therapies. Uh, and interestingly, this can occur below age 40. And the famous example for this one is the Hollywood actor Michael J. Fox. Early on, I think he's within 30, in his 30s, he has actually had symptoms of Parkinson's disease. And he has funded a lot of neurobiology research in the last 25 to 30 years. It's more common in males, 63% compared to females, only 37%. So it's a reverse order compared to Alzheimer's disease. Common in farmers, especially farmers who use herbicides and pesticides, usually have more incidence of the Parkinson's disease compared to normal people. So it's genetically, it's very rare disease, less than 10% hereditary cases. Next one, please. So the pharma companies are still trying to come up with different target-based drugs for Parkinson's disease. On the opicapone is the another catechol O-methyltransferase inhibitor that works to somewhat extent. But all these drugs, sulfonamide, they're all used as a adjunct therapies along with levodopa, carbidopa, and other drugs that people are using. Interestingly, Norinas is also adenosine A2A receptor antagonist, used also as an adjunctive therapy in combination with levodopa, etc. So several other drugs for symptoms like excessive drooling are also available, but it's just only symptoms. They're not actually not these drugs do not prevent the Parkinson's disease progression, but they prevent some of the symptoms of the Parkinson's disease. Next one. Pathophysiology of the amyotrophic lateral sclerosis, people call it ALS for the short. Vascular endothelial growth factor is the primary cause. I mean, there's the ALS2 gene that gets mutated and which causes the problems in the capillary density in the spinal cord. And that causes this neurodegenerative disease. Next one, please. So, very rare disease, maybe 2 in 100,000 people, and so far there are no known cures. Often requires a lab test or imaging, and ages above 40 or above have progressive degenerative disorder. And of course, people diagnose it with ALS2 gene mutation technique. Next one. So let me come back to the drug discovery part of it. Yes, you have all these diseases, especially neurodegenerative diseases. The success rate is very, very poor compared to if you look at cardiovascular diseases or diabetes or other diseases, many other diseases. We have several drugs, but for neurodegenerative diseases, we do not have much of success in bringing drugs into the market. Typically in pharma company, the way we proceed for drug discovery is that, the any drug that is approved for human use must be safe and it should have good efficacy in treating the disease. Uh, so when we do it, many drugs either they may work, but they're very toxic. In such cases, we cannot really give them to the patient. Yeah, in animal models or my in vitro models, drug is very efficacious, but when you put into the humans, they're very toxic. So those kind of drugs do not make it in the market. So here in this case, red light, I've seen in the balance that risk is greater than benefit. So you do not want to take those drugs into the market. On the other side, left side, you say green light, where is actually benefit is much higher compared to the risk or safety issues. In such cases, you actually take these drugs into the market. So basically, you need to find a balance between the efficacy and safety. That is what the entire drug discovery and development process boils down to. Whether a drug biologically it works or not is not the issue. It has to be safe enough, but works very well for the patient. Next one, please. So when we do early drug discovery, we do a lot of screening approaches. In the screening approaches that we have, I'm not going to talk about all these different screens but what we do is we put it in green, yellow, and red chart and say, hey, we have more greens, we can proceed into the clinical trials. We have more reds, we cannot proceed into the clinical trials. We have a combination of yellow, green, yeah, maybe we can carefully move into the clinical trials. So this is kind of decision tree we do with a lot of different screens that we use in drug discovery and early preclinical development, that we take molecules into the clinical development with a cautious approach. Next one, please. So if you look at the diseases in the world, Alzheimer's disease and other dementias are accounting for about number of deaths. In 2015, at least 1.5 million. Of course, the major one is ischemic heart disease or coronary heart disease, the major deaths, but Alzheimer's disease also contributes to a significant deaths in the world. Next one, please. So this is the funnel of the drug discovery. What we do is our chemists, medicinal chemists, look at the structure of the target, and they have a three-dimensional crystal structure of the protein or enzyme or receptor, whatever it is, and they try to screen with the bioinformatics a lot of drug combinations, chemicals, and see which ones fit into the active site. From there, we come down to about maybe 10,000 to 20,000 compound candidate drugs, and we actually do the lead optimization with several screens. And finally, we come down into the ADMET properties, drug metabolism, toxicology studies in animals, etc., and then take into the clinical trials and step in. So out of 10,000 to 20,000 drugs, you get only one drug into the market, and it may take about 10 to 15 years. And for one drug to get into the market, it may take $2.5 billion. So costly. In India, we cannot afford it because that kind of money we cannot put into the drug discovery and development. So most of the drug companies in India abandoned the drug discovery and development research, primarily because it's becoming cost prohibitive. Beautiful research excellent research is being done in India, but we cannot take it further because of we don't have that money to put into the research. Next one, please. So, but the problem is, we do all that thing. And if you look at the FDA approved drugs, I mean, I don't see much of difference in how many drugs are approved by FDA since 1996. I have the chart here. In 1996, there were 53 drugs were approved. But you look at until 2017, there are either 20 or 30 or 40 drugs were approved. Only in 2018 that we reached back to the 59. Again, we are dropping down to 48, 46, or even 20s. So the problem is that it's very poorly productive outcomes out of 10,000 to 20,000 molecules. We are not getting more drugs into the market rather than what we got in 1996. We still are the same in 2021 today, the number of successes by US FDA approvals. The part of the reason is that because we assume clinical trials can be translated by animal models. Unfortunately, that is not happening, which is why the reason the models and paradigms are changing by pharma companies today to improve the productivity of success. And in fact, genomics, proteomics, molecular biology, molecular, transgenics, knockout animals, everything we applied, in vitro heterologous expression systems, you single cell, what is that, flow cytometry, genomics. Techniques wise, we don't have any problem. We have tremendous technology that improved in the last 30 years, but our productivity still remained the same. And so what the problem is, it's not the technology that we are inventing, it's the problem with the model that we are choosing. And that is where I'm getting to in the further discussions. Next one, please. I don't need to. So some people are actually moving away from small molecules and slowly moving into biologic molecules, more protein type of molecules or peptide type of molecules. And you can see that slowly, the more and more biologics are also getting approved by FDA in addition to the small molecules. Next one, please. Same thing. Now people are more focusing on orphan diseases, which means orphan diseases means by definition only less than 200,000 people are there, patients are there in the world. That means it's very small population. Unlike heart disease, there are millions and millions of patients are out there, but only 200,000 patients worldwide. That means if I have to really look for the patient to do a clinical trial, searching for the patient is going to be very difficult for me even before I can do the clinical trial. But even orphan drug research is very much on the rise now. People are spending more money, and in fact, $339 billion has already gone into orphan drug development. Next one. So if you look at I've been in 30 years in industry, I've been watching the paradigms, how they are changing. Back in 1960s to 1980s, it was just you dose the animals the drug, and if it is safe and it has some preclinical biology, pharmacology effects, efficacy-wise, you just move it into the clinic. And suddenly, when you go to the clinic, you realize that the drugs are actually not getting absorbed into the circulation, unless it is IV administered or IP administered. But most of the drugs that we are developing are oral drugs. The reason for that is because oral, you can take it at home every day. That's easy way for a patient to take medicine. And the other thing is IV and all, you need a hospitalization. You have to go to the doctor or hospital every day. And so that's not going to work. So when you look at this, oral drugs, efficacy is only 36% of the drugs have in the clinic. But vast majority of them actually failed in that. About 39% of the preclinical pharmacokinetics. And so small toxicities. But if you look into the 1990s, efficacy part of it didn't change in the clinical trials, but toxicity has gone up by 43%. It's just you're shifting from one thing to another thing. Now, pharmacokinetics was the major failures in 1960s to 80s. But when you come into the 1990s, your failures, because you're having more drug getting absorbed into the systemic circulation, you have toxicity failures because the drug is becoming toxic. And although it may be efficacious, but the reason you want to drop the molecules because they're very toxic. And then we went into we also put a lot of screens for toxicity evaluations, in vitro, in vivo, everything. And then when we came into 2000s, in reality, all the drugs go into the phase 3 trials. 85 to 90% of them do not make it into the market. Less than 10% of the phase 3 drugs make into the market. All those drugs fail because there is lack of efficacy. So when you do a preclinical biology, pharmacology experiment in animal model early on in drug discovery, and then you wait until phase 3 to see the efficacy, you spent out of $2.5 billion, you already spent $1.5 to $2 billion just to realize that your animal model is not translating in terms of the efficacy. So this is the risk that we need to take in drug discovery and development. Next one, please. Next slide. Yeah. So the problem here is that it's not the lack of ideas, the lack of depth of technologies and everything. We are underestimating the complexity of the human biology. We know that we have about 20,000 genes in humans. Not all genes are expressed in all tissues. For example, in lung doesn't have the same genes that are expressed as in liver. If they both express the same genes, lung and liver look the same morphologically the same, but they don't. That's the reason, because they express totally different genes. And inter and intra-ethnic genetic polymorphisms of many genes. In fact, Caucasians versus Africans, and even within Caucasians, some genes are polymorphically expressed. That means certain 10% of the patients either don't express the gene or have a mutation in the gene. So all this boils down to the human biology. And now, as we age, in fact, we put on more mutations due to the environmental or lifestyle factors that we are living with. And I don't know the actual number. They think that there may be more than 1 million proteins, and many of them are post-translational modifications. Many times family of proteins work together, not a single enzyme. And epigenome, I can't even find out what is epigenome anymore, because it's like a space map. And you look at the Google map and you forget it. You don't know where you are putting your transcriptional and translational factors into that. And the metabolome. Metabolome is the phenotype. But again, there are only 7,000 metabolites that in circulation that you can see. And you need very, very sophisticated, sensitive technologies to identify many of those metabolites. And that is one of the reasons you don't understand metabolism as well, all these factors. So genome to phenome, there's a lot of gap in between that we can identify. Yeah, you can identify a genotype, but you don't know whether it translates into the phenotype or not. Next one. So the complexity is that if you give an oral drug, right, in the intestine, there's an intestinal barrier called, there's a P-glycoprotein, one of the drug transporters, very well studied. That's a gatekeeper. It's like a Gurkha. It sits there and doesn't want any foreign molecule to go into your body, and it prevents it. And that's the reason many of the drugs we try fail in early drug development or early drug discovery. And that is one of the barriers. Then the drug gets into the liver, somehow manages to get into the liver. And in the liver, liver is a screen. It's a detoxification organ. Anything foreign, it wants to throw it out of the body. What it does is it puts a lipophilic, hello? Lipophilic molecules become hydrophilic, and either they get excreted through the kidney, or they're also excreted via bile with the bile transporters, etc., etc. So liver is a sink to throw the molecules out of your body. So the drugs face all these barriers, and then you go into the target organs like brain. There's already Dr. Naren said there's a blood-brain barrier that also prevents your drugs to go into the brain. So for neurodegenerative diseases, then we need to put all these things together if you give an oral drug: intestinal barrier, hepatic barrier, and blood-brain barrier. So we need to look at all these together in a dynamic fashion to create a model for us to understand. Next one. So drug metabolism. The problem with drug metabolism is not just an excretion part of it. So a lot of drugs and environmental pollutants are metabolized by the enzymes called cytochromes P450 in the liver. And in fact, people above 55 to 60, they have many diseases. I mean, like heart disease, arthritis, and diabetes. And so they're probably popping up 5 to 6 drugs at a time. And on top of it, my Ayurveda guy gives me another Ayurveda medicine, and homeopathy guy gives me another homeopathy medicine. I'm taking about 10 drugs or medicines, and all these things are like atom bombs. They interact with each other. Drugs, drug herbs, and especially in cytochrome P450 level or drug transporter level, these become very toxic. And we don't know. Many people die of uh, for some reasons. Hey, he suddenly his heart stopped. It could be drug-drug interaction. We don't even have in India the system that how to monitor a patient why he died. And these are all the problems. That we also have some drugs are getting metabolized to very toxic reactive intermediates. They covalently bind to the proteins or DNA and cause either immunotoxicity or cancer. So it's a very complicated process of the liver detoxification process. We assume it is, but it also puts toxic metabolites into our system. Next one. So this is just a standard slide of the pharmacokinetics. Uh, I mean, you give a drug orally and you look at how much drug is present over the time for 24 hours. We measure the half-life of the drug, and typically the ideal half-life of the oral drug should be about 8 hours. And we also calculate bioavailability. We just give an IV administration and we measure the oral administration AUC or area under the curve, and we say, hey, only 30% of the drug is absorbed compared to IV administration. So we know, is it 10% absorbed by oral or 5% absorbed by oral, and we look into that in more detail. And in fact, it's not straightforward. If you look at just, I'm giving you for comparison, males and females at the bottom of the chart have a different pharmacokinetic properties, especially in terms of the clearance and volume of distribution. So gender differences are there, ethnic differences are there. A lot of other factors do contribute for these differences. Next one, please. So we have in this process called linear and non-linear pharmacokinetics of drugs after oral administration. Many people think that any concentration of anything would be fine. That's wrong, because biology also has saturation processes. Drug metabolism enzymes get saturated, drug transporters get saturated. So you do get nonlinear pharmacokinetic observations. When a drug has quite a bit of nonlinear pharmacokinetics, it's very difficult to do the regulatory drug development on those kind of drugs. Next one, please. So drug transporters. Right now, we don't know how many transporters are there. I'm just putting up here that the liver, intestine, and kidney for now. I didn't put didn't brain also. All the transporters are gatekeepers here in these different organs. So if a drug binds to a drug transporter, either it can be taken up or it can be thrown out of the cell, right? So these complex mechanisms also regulate how much drug goes into the body or your target organ. And this is right now, FDA has got a lot of guidances on these human drug transporters. We need to study only by in vitro mechanisms because before we go into the clinical trials, we don't have any data on the human drug transporters, human drug metabolism. So we use in vitro models mostly to understand these things. And in fact, I'll come to the later to the slides. These transporters of the humans are totally different from the animals. Next slide, please. So here I'm just showing that intra-ethnic and inter-ethnic variations on one of the drug metabolism enzymes called CYP26. And previously in 1990s, and if a drug is metabolized by cytochrome P450 10% of the Caucasians do not have this gene, it's deleted gene, or 17% of Germans actually do have multiple copies of the same gene. Or if you go to Asian, 20% of them don't have this gene. So it's very complicated if you go to inter-ethnic and intra-ethnic differences. Even in Indians have a lot of differences. So if I do a clinical trial in the US for their Caucasian population, if I bring the drug into India, I need to do a clinical trial separately for Indian population. It's not so easy that, okay, that I have a Caucasian data in the US, I'll just use that data here in India. It doesn't work for you. The drug may kill you, actually, in fact, because of the polymorphisms we have in drug metabolism and drug transport. Next one. So the other problem is sometimes, as I said, these are all adaptive response systems. So cytochromes P450, especially in the liver, they get induced, which means if I take a drug for 3 to 4 days, it's perfectly working okay for 3 to 4 days for me. After maybe 2 weeks, it stopped working for me. It's like antimicrobial resistance, right? You send the antimicrobial drug for a while, bacteria become resistant. Same thing with anti-cancer drug. If you take it for a while, the cancer cell becomes resistant to the drug. So same way, our body is equipped with these resistance mechanisms. It's evolutionary, that is actually a very good thing to protect us, but for drug discovery, it's becoming a problem for us. For example, there is a drug that goes into the cytosolic fraction, right? There's a nuclear receptor called pregnane X receptor, and it forms a hybrid with retinoid X receptor. And that PRE actually goes into the nucleus and activates the gene, and the same gene that metabolizes the drug gets activated and transcriptionally activated, and more enzyme is produced. So more drug is metabolized. So after 2 weeks, your drug is not working anymore because the drug is not there. It is metabolized and thrown out of the body, right? So these are all the things that we need to look into when we come in with the drug discovery combinations. And in fact, even herbal preparations do the same thing. So I'm talking about some of the grapefruit juice compounds and coumarins and other compounds. They do the same thing. They activate these genes also. So our phenotype is different, although genotype may be is the same from all of us. Next one, please. The other downside, you probably are hearing about COVID, right? COVID, a lot of people are giving hydroxychloroquine, ivermectin, and many other drugs for the COVID treatment. In reality, COVID is a disease of the cytokine storm and inflammation, right? And if I have a drug that is metabolized by one of the cytochromes P450s, inflammation actually downregulates these cytochrome P450 genes. So my drug is not metabolized compared to the healthy individuals, which is highly metabolized. So healthy individuals are different from diseased individuals in this particular model. So hydroxychloroquine normally in healthy subjects has a 22 days of half-life. Typically, as a drug development person, I will not take that molecule into the clinical development because I don't want something to sit in my body for 22 days after a single dose. You don't know what it is going to do, what kind of toxicities it does. Does it cause cancer in 10 years? So if you're giving the same drug for 5 days to 10 days, you will be able to see it even after 1 year, the drug in your body, even if you stop it giving after 10 days. So I don't want those kind of drugs in my body. Okay, so FDA regulates all these parameters and say, hey, your drug has a 22 days half-life. I don't want you to take into the humans, period, right? So all these things that do put a more pressure on us how to develop a drug. Next one. And the fun starts, you really work very hard for 14 years, 15 years, you put out a drug into the market. Now, sometimes the fun really starts here. There are some drugs, like for example, troglodyone, it was an anti-diabetic drug. That was wonderful anti-diabetic drug. Within 1 year, it was in the market. Probably about 6,000 to 7,000 people died. I don't remember the numbers. People died. The problem with this is that when you do a clinical trials, you have a very small population. Phase 3 trials, maybe 3,000, 4,000 people. And when you take the drug into the market and people start taking it, millions of people are taking it. Now you'll see post-marketing reactions. So 1 in 5,000 people dying, you cannot detect in the phase 3 clinical trials. I cannot do a clinical trial with a 10,000 patients because it's very costly. As such, I'm spending $2.5 billion. I cannot spend any more money to do the phase 3 trials with 10,000 population. So these are the problems that we face, and the drugs, many of the drugs that go into the market, now they're withdrawn from the market. And this is one major issue that that we face. Once the drug is withdrawn from the market, the, especially in the US and other developed countries, the families of the patients start suing the pharma companies. So it's a huge risky business. I spent $2.5 billion to make a drug into the market, and now I have $5 billion lawsuits. Where do I get the money to pay for those lawsuits as a pharma company? So the pharma companies are very, very, very cautious in discovering and developing drugs. And even regulatory agencies like FDA, EMA are very, very restrictive, are conservative in approving drugs into the market. Next one, please. So, next one. Next, oh yeah. So the other problem is sometimes cardiac toxicity. Many drugs we don't know. Actually, what it does is a drug, especially through drug-drug interaction. I said hydroxychloroquine in COVID. In fact, they're also given azithromycin and another antibiotic. Azithromycin increases the cardiac toxicity. So hydroxychloroquine inhibits the azithromycin metabolism. Azithromycin goes to a toxic concentrations and stops the heartbeats. Basically, that's it. It stops the potassium channels, okay, and heart stops and people die. So for that reason, there are lots of drugs are withdrawn from the market due to drug-drug interactions. Next one. So the other issue is that now people are trying to do the genetic drug discovery. I mean, this is another fun that is going on, gene therapies. Autism is one of the neurodegenerative disorders. When the kids are born with autism, they don't survive more than 50. Many of them don't survive. And in recent years, with exosome and endosome gene sequencing technologies, they identified about 800 gene mutations are responsible for the autism. So how are you going to diagnose autism with the genetic technologies? It's a big question. Same thing with diabetes today, that we have more than 200 gene mutations are identified in type 2 diabetes. So that's why it's becoming very difficult to treat every patient. Next one. So we are coming to another drug discovery paradigm shift. The problem is it's the human biology that we don't know. We know a lot of animal biology that's really not helping us to design drugs. Yeah, I can, if I'm developing drugs for animals, yes, I can do animal biology, perfectly fine. But I'm developing drugs for humans here. So I need human biology, not animal biology model data. That excellent work is being done in animals, but that's not helping me in discovering new drugs and developing new drugs. So the paradigm shift here is that I actually spent 3 to 4 years while I was a regional head at J&J in New Jersey, that there were some models of 3D microfluidic chips, organ-on-a chips, etc. I spent 3 to 4 years. My team actually worked on it. We got a prototype chip on it. But again, these kind of models take a long time to develop. In India, I actually became the expert committee member at ICMR to push the alternatives to animals research, primarily to do the human 3D models more than animal models. It's not just for ethical reasons, but also for scientific reasons. So we are developing molecular diagnostics tools with whole genomic sequencing, identification of gene mutations in disease earlier than later, non-animal human 3D organs, microfluidics, 3D bioprinting methods. We've developed lung model, 3D vascular lung model. We have 3 drugs that we have already identified for the COVID. One is in clinical trials. And we also have a leaky gut model, human leaky gut model with a 3D bioprinted model. and that we are applying for alcoholic liver disease. We have a few others that are coming up. And of course, I need to, maybe with Naren or somebody, we need to come up with the 3D neuronal models. If we and that way can collaborate with each other. So the other thing is the stratifying the clinical trials with molecular markers, biomarkers you really need as soon as you go into drug discovery. Because in humans, unlike in animals, you cannot open them up and see if the drug is causing liver toxicity or lung toxicity. So you really need real good biomarkers to look at into the human safety issues and also efficacy. You need biomarkers. So drug repurposing. The thing is that we already have 8,000 FDA drugs. I don't need more and more drugs. And out of these 8,000 FDA drugs, if I can repurpose in the human 3D models and see if I can identify many other drugs that may work for human diseases, other human diseases like Alzheimer's disease or Parkinson's disease, etc., and we can actually make a huge difference. Roadblocks, regulatory is too slow. Still, regulatory agencies still recommend animal models. And for that reason, pharma companies don't have a choice. They have to do the animal trials before they take their molecules into the clinical trials. This is where regulatory interactions are very essential. When I'm developing these 3D models, I have to convince regulatory agencies that you need to use these models, not animal models, to go to the clinic. And I need a lot of data to clinical validation data for it. And so that means I need a lot of funding to also to do it. So there's already a couple of companies, especially Emulate, a company from New York, working with FDA with some of their liver 3D models. So regulatory is also looking for these models, whether they can mandate these models more than the animal models. So in 2025, FDA wants real good models so that they can start mandating these 3D models instead of animal models. EP, Environmental Protection Agency, said by 2030, they want to get away with all the animal models they're using for their toxicity evaluations. So we are going ahead and we do need a lot of collaboration. What the problem I see in India, I mean, there's nothing I'm talking about is a complaint, but when I worked at J&J, I had access to MIT scientists, Harvard scientists, Princeton scientists. I had a lot of academic industry collaborations to make these things happen. Because in drug industry, we are supposed to bring the molecules out into the market. We can't do academic research. But the academic researchers have to work with industry to be able to make this happen, more drugs. So you need regulatory, academic, industry interface happening every day. We need to nurture that kind of a culture, even in India with IISc, IITs, AIMS, and all these types of institutes. If we do that, India can become a great hub of drug discovery innovation in the future. Next one, please. So the goal is to develop a multi-organ chip. We have a 3-organ chip. I can't present the data because we have some patent and confidentiality issues. So I'm not presenting any of my data at the moment. Sorry for that. If you want, I can privately talk to you with a confidentiality agreement with those models. We already have a lung model. Some people have already developed a liver model. And we have a gut model that we're already in the process of building. We're still waiting for kidney, brain, or the major models that we need to incorporate into the system. And we want to create these multiple organ models. See, blood goes into the system and blood keeps flowing from one organ to another organ. This is very, very crucial and critical for the drugs, metabolites, or even the endogenous metabolites, fatty acids, proteins, amino acids. All these things get bathed in tons of the biological milieu receptors, etc. So you do need to create that kind of a model. I cannot take a single cell, put it on a petri dish, and try to study in the absence of the rest of the human body, right? Same thing, I cannot just only delete one gene in animal and say, hey, and put a human gene in place of it, but the rest of the mouse is not human, it's mouse or rat. So how can I study the entire gamut of dynamics that's happening in the humans in an animal model which is only deleted with one gene? So this is the problem, that we don't have good animal models to reproduce the human safety and efficacy issues. So the only alternative is to build these kind of models, and we can also do pharmacokinetics, we can do histopathology, you can do multiple dosing studies with this microfluidic chips on humans. These are all called zombies. I mean, when I presented it in 2004 to some investors, and they looked at it and, hey, these are zombies that you're creating. And that's what we are essentially creating here, zombies. Next one, please. So for autism, etc., I mean, we are right now doing a little bit of it. You can take the autistic patient's cells or wild type cells. I mean, you can actually, as he described, you can take stem cells and you can differentiate them into glutaminergic neurons or dopaminergic neurons or something like that. And you can actually study the neuroscience, these models. What we can do is create these models, pack them with endothelial cells and all the other barriers that we have, create a 3D barrier and bioinks, which makes it physiologically the same, almost not, I would say same, almost compatible to understand the human biology in the brain as it happens. Mouse brain doesn't provide you the opportunity to study the human biology in more detail, or the progressive neurodegeneration. Next one, please. Next slide.